So let me discuss about today's topic, light sheet microscopy. First of all, majority of this technique is using a fluorescence mode. So sometimes we call it light sheet fluorescence microscopy. So in Nature Methods in 2014, that was method of the year. It was chosen as a light sheet fluorescence microscopy. So why it become an emerging recent new technology which grabs attention for all the world biologists, because its ability to make three-dimensional imaging, high speed and low toxicity. So I want to start with you to show actual image. This is just an exquisite image which is unimaginable in previous, even like 20 years ago, this is unimaginable. You see how detailed that this is, like neurons in the spinal cord and the cellular detail in 3D. And this is just amazing resolution in 3D. So I want to show you a movie which actually shows the power of this light sheet technique. Of course, it came out in 3 years ago in Science cover, Resolution Revolution. So that means this is real cutting edge. And actually some of my students in our lab is developing also a new type of light sheet microscopy too. So let's take a look. This is a Nobel laureates group's work. You see, it's clear by adaptive optics, and these blue dots are kind of sugar molecule, and the immune cells are coming in and uptake this dextran. So you can see this one's the dynamic cells come in and out, and this is an inner ear. So you can see these cells actually engulf. And also you can see this three-dimensional nature, and this is kind of blood vessel. You see RBCs are actually coming in and out. You can see this blob. These are like penetrating RBCs, and you can see the cells are moving in and out. Immune cells, pressure valve, mitotic cells, blood flow. This is like real tiny sample, but super high resolution for 3D in a living body. And this is just an amazing thing for all biologists to look into. And you can use your image processing tool to specifically take out the cell and their three-dimensional trajectories. And these color code, you can also calculate the velocity. So what it means is this immune cell migration through zebrafish inner ear, and they are picking up sugar particle and they're moving. This is now what it means is, before we didn't have the ability to see this. However, now biologists or scientists can ask a lots of questions about what's happening from here. So this, I would say, like, this is just fascinating. I think this will make us to understand at the cellular dynamics much more better, and even in intact in vivo condition. Okay, so now then what we've seen, so we have a question, how can you achieve that? What's the concept of this new type of microscope called light sheet microscopy? So there comes one name here, so-called SPIM. Sounds interesting when you selective plane illumination microscopy. You can imagine what does this mean. So that concept is a little bit different from what we have done before, because you can see now there's a detection objective lens. You can image that fluorescence, but the illumination is now a little different. It's actually decoupled. Okay, what it means is illumination is now come from outside on the side excitation, and the shape is not the same as conventional. It make a sheet of light, like a sheet. You make light as a sheet to cut through the sample, and then you take an image. And of course, there are various ways to make light sheet, but let's think about what's the practical issues when you take a conventional imaging versus light sheet microscope. So the SPIM, one example of this is called SPIM. and you are actually cutting through with illumination. But let's think about confocal microscopy. Confocal uses the objective to shine light through. So when you are scanning, you are actually illuminating this much through the sample. While SPIM, you are illuminating this blue light sheet, and you are actually taking that all together with a wide field detection. So the position of the sample which is illuminated are now just directly imaged. But in confocal, you all know out of focus, above and below, it is being shined, but will not be detected. So that's wasted illumination. So that causes toxicity, I mean, photo bleaching more. So compared to confocal, this has something reduced toxicity you can actually imagine. So now, okay, so how can we develop and implement? So because this class, we are interested in not only like how things work, how can you build it? So let's make a simple setup of how to make it. So you can see there's an orthogonal condition of detection and illumination. So detection, you can generate a conventional 4F system of microscope microscope here, actually this one, so that you have turbulent, you have CCD 1, 2 for multicolor detection, and excitation, you are actually shining from the other aspects of light sheet. So it's actually quite simple, in fact. It looks simple while making it is different. Or please show your face, those who are registered students. Okay, so let me ask a question to Jungmyo. So you're shining with a laser light sheet. What does that mean? Now you are for three-dimensional imaging. Consider like confocal or two-photon microscope. You are not, you are making the sample, you are actually cutting through with a sheet of light excitation. So you are cutting with a light optically. So optical,
section.
Yes, sectioning of a sample with your light sheet, right? And interestingly, this is decoupling the illumination and tell me again.
Detection.
Exactly. Illumination and detections are now decoupled. This is very important aspect, both good and bad. A bad aspect is alignment might not be as easy. Good aspect is you have more freedom. Okay. And this illumination light sheet has about 1 to 10 micron thickness. And in fact, this one exactly means the actual resolution of your detection, because now we are getting into real three-dimensional imaging. Okay, so this one will be comparable to the depth of field of typical objective lens, in fact. And why this become a faster imaging? Because you are using wide field detection, not like point by point scanning, such as confocal or two-photon microscope. So you are taking entire 2D field of view with just a single shot. So how can we achieve 3D imaging? Hey, you have a sample, you're cut through. How to get 3D imaging? You either move the sample ups and down, taking image, or you can move the sheets ups and down. So that's it. 3D image by moving a sample of light sheet in the axial axis. But as I described here, this has a lot of very important advantage compared to confocal microscope or wide field microscope because it has minimal photo. Let's see, tell me again.
Bleaching.
Exactly. Because you are only photo bleaching happens only where the excitation light is. Okay, so let's move on to then who actually made this a cool invention. Many times you think that's a new invention, but many times, actually most of the time, I think 100 years ago, there's always people who have thought it long time, even before the invention of a laser. So here comes a chemistry. It's actually about almost 100 years old. There's a Nobel Prize, not exactly the invention of light sheet microscope, but this guy actually used it for the first time. So how does it look? Remember that time there's no laser. So he made a light coming in and using lens to shine. But look at that. Now you are shining on the side of the sample. In fact, he uses a slit to make this very narrow too. And then you observe orthogonally. Yes, this is an arrangement, a new type of imaging, because he was doing colloidal experiment and colloids it everywhere. So when you take a picture, all above and below, there are all sorts of this colloid particle scattering light, giving you a blurred image. So you cannot really see. So his invention is making a light sheet with it. So he named it that. In fact, it's a slit because he had used the slit to make a light sheet. He called it that ultra microscope. In fact, this is one name of commercial microscope nowadays for this light sheet. And his name is Zsigmondy. And, you can read his Nobel lecture in chemistry on colloidal chemistry. And here's a picture of actual system what he made. So there's a microscope. These are, at the time, there's no laser, so not easy. But he made this nice emergent ultra microscope. You know what? If I look at his Nobel lecture, he put his image with this microscope. Look at that. This is the image. Very, very poor image though. But consider this 100 years ago. This is scattered image of a jelly flakes. So scattered one, it's like one way of dark field microscope as well, because this dark part is an empty water space. But we have to wait. The humans wait for about almost 100 years, and now first application we catch up that to utilize this to a biological specimen. In this case, guinea pig cochlea by Vui et al. So that's 1993. So look at this nice cross-sectional image of inner ear, which you guys kind of know already, right? Organ of Corti and how this inner ear vibration gives us this frequency response so that we can understand and perceive a sound and process. So this is actual image of light. But take a look. You can see a fine line over here. This is a kind of an artifact and which we have to overcome. But remember, this is the kind of recently the first time. And then what happened? I was a graduate student since 2001 when I was at the Golden Conference, Golden Research Conference. And there's a person who came in who is a little chubby person. And he was, to me, it was a little rude at the time. I was a graduate student. He talked to the conference chair. Hey, I brought a very cool thing. I have to present this one. Like, whoa, it wasn't on the schedule, but he let him to present. You know what that was? That was actually selective plane illumination microscope. And his name is a famous E.T.Z. Stelcher. And he's the postdoc advisor of Nobel laureate Stefan Herr. And he's a kind of a very interesting, creative person who made a new history of light field, light sheet microscope. So that's the first one. The first author is Jan Huisken, who's like a big picture. And this is so first invention of spin. And just a few years after, so this is came out in Science, and 4 years after, there's a Nature Method by Philip Keller, who made it a little different version called digitally scanned light sheet, so that you can name it DSLM. DSLM. And since then, like there was a boost of, hey, this may solve many of the interesting unresolved issues. So 2014 by Eric Betchik, who's a Nobel laureate from a super resolution. And that group shows light sheet with lattice, which means there's a light shift, but you have to take pictures like sequentially, but he made like this one as a lattice, so multiple light shifts simultaneously. So that has been approved and shown, and the first commercial light field microscope was just done. You see the timing, it's just so quick. It has been commercialized very quick, still a little expensive, but people who want to use it can use it. So this is the first commercial one, JICE one. Actually, this system is within the GIST. Just next building, you can just sign up and you can bring your sample. You can actually take three-dimensional images. All right, so before we get into the detail, why don't we spend some time like, hey, what can we do with this? Okay, what are the applications? So what made this very revolutionary is the embryogenesis. Drosophila, which is like fruit fly, zebrafish. Lucky thing about zebrafish is young zebrafishes are transparent almost. So it's easier target for light microscope and other organisms. So let me show you one picture. So these are embryonic development. You know, developmental biologists are always interested in which cell goes to which cell forms a nervous system, which cell goes into differentiate into eyes. How does this happen? But unfortunately, you cannot see all the things because it's rather big and you don't have a nice 3D imaging modality for all directional aspects of good isotropic resolution. Look at this. So this is now possible even in a living system. So we call this as a, this is a little different. In total means as a whole, like in vivo imaging. This is in total imaging, right? And now if the sample is transparent, you can have a imaging with a very fast cellular dynamics of embryos and small organisms. Also, some of you are maybe working on like three-dimensional organoid because that's a little more difficult to image compared to 2D or cell culture. If it's a 3D, it's more difficult, so-called spheroid. So this is, you can see like a line of this. On the side, you can actually see a cut-through view, and this is a cut-through of 3D organoid. So Laxman, so can you guess what kind of this cell culture is? Spheroid tissue culture, but it's not 2D, but what kind of?
3D?
Yes, 3D is very good for this. And not only with that, almost transparent marine organisms such as like this, squid, plankton, flatworms. And this technology, the good aspect is low toxicity, phototoxicity or photobleaching that allows, and the other is wide field detection, so makes it faster. So that allows us to get into functional imaging because functional imaging is usually requires a more dynamic and faster imaging. So even small animal brain activities. So there were nature methods which just revolutionized the whole brain of zebrafish. Now this one has a calcium indicator, but you can look at this, the whole brain activity you can now actually visualize. So while doing the zebrafish showing up pictures of this, we don't really understand properly, but you can see a blinking of this whole neural networks. So this is just a fascinating whole world about this functional fast imaging potential. And hey, wait a second, what about structural imaging? Because we suffer from point-by-point scanning nature for, let's say, 2-photon microscope to see the whole three-dimensional brain. It just takes too much time. But light sheet can make it faster because it's a wide field detection. So even structural imaging, it's good. And now the advent of tissue clearing technology, we actually can see fixed and cleared image. So this is an example like came out last year. The whole brain tissue cleared. You can use light sheet to take pictures of it. And the right one is actually a whole three-dimensional blood vessel distribution in a heart. Okay, So these are something really cool thing that enabled us after we start to use light sheet microscopy. Then the question is, okay, that's good. So how can you make sheet of light in space? We know some optics, so it's not, not impossible, but we have to know a little more detail. So how to make light sheet? You have a lens, then lens make a light to focus in one side very thin, but the other side, your lens do not have any curvature, then the other side just the light penetration is the same. So that's the trick. So that's how you can make static light sheet. But remember, to make this a long sheet, you do need a very low numerical aperture or low angle incidence. And usually this one is coming from laser. So laser profile, intense profile is usually Gaussian. So one way is you can use a cylindrical lens to create this. The other is just make a low NA lens, then you can have a nice, long, like needle-like beam, a little thinner waist at the middle, and you can effectively just move up and down very fast. Then effectively, you can make a kind of light sheet. So that's how you can make it. But that has been done, so-called DSLM, digitally scanned. So people also want to do like 2-photon waves. So of course, people done. Scott Fraser group, originally at Caltech, now at the USC, he created 2-photon SPIM, selective plane illumination microscope. Okay, that sounds good. And then another thing is a Bessel beam, which I'll explain soon, which the shape looks more like uniform compared to this. Okay. Then question is, okay, what is Bessel beam? Interestingly, you can look at this one. This one has a much thinner, so this is really good. This is done by Eric Betchik's group. And so this is what you saw, a very nice the movie I showed you how to make it. And this Bessel beam has a very interesting profile. The nice thing is this is thin, and over the axial direction, the thickness doesn't change, while the others are all kind of changing. But the downside is, you see, there are like side lobes, which causes an artifact. And just using a Bessel beam has suffers from big side lobe. However, there's a trick. If you use 2-photon excitation, the side lobe can decrease. And the reason is 2-photon excitation, the intensity outcome is square of the illumination. So the fluorescence outcome is square of the illumination. That's what 2-photon nonlinearity actually causes, and that suppresses this side lobe. So let me go back again. So typical laser, you have a low NA objective, you shine it. That is called, let me see, 수현. How do we call this profile? Exactly, Gaussian beam. So this one and these one are Gaussian beam. And the next is the concept how to make beams very uniform. You have to consider like interference from basic optics concept. When you generate interference pattern, you shine the light, coherent light in oblique angle, then you will generate this nice interference pattern. Center is high, but the other has also equidistance. So this Z profile is almost sinusoidal. Hey, wait a second. Can you make that by illuminating 2 beams, one this and the other this direction in space? Then circularly you can make this profile, and that is called Bessel beam. So you can see from here, Bessel beam is nice because over this axial direction, the size doesn't change because this is just a simple interference of oblique beams. But you can see the side lobes are prominent, so that causes a problem and artifact. That's why Eric Betchik's group, this one square it to create 2-photon Bessel beam. so that to suppress this side lobe. So hopefully that gives you an idea of how to produce a sheet. One way is just use a conventional cylindrical lens, and the other is actually Gaussian laser beam, and you move very fast up and down to create a sheet of light. And there's another trick of even make it smaller uniform by use of Bessel beam. Of course, there are other techniques recently you can actually see. So the question is for those biologists, hey, I've learned about wide field we have in the lab, we have confocal microscope. Why should I care about this? So comparing with the laser scanning, confocal, or even two-photon microscope, and you can find out some niche application for this. So when the sample is sick, confocal doesn't work very well. And you know that confocal and two-photon all has a suffering from axial and lateral resolution mismatch. So it looks like a little weird, especially like embryogenesis, which you want to see all different directional resolution to be nice. So all side, this is something light sheet can do a good job. And long-term live imaging, you know that confocal suffers from a lot of photo bleaching, which is detrimental. And two-photon case also, this is good because it only excites one point, but that point you may use a too high intensity, then it can also give a moderate photo bleaching. Okay, and you have to move it one by one, so it's faster imaging is not that great. So conventional scanning, point scanning just takes too long time for some application. And that's why when we have to consider light sheet. But remember, light sheet doesn't necessarily give a super resolution. So that's kind of a limitation. So here comes the comparison. So when and why we use in terms of photo bleaching and imaging speed. So you guys review confocal microscopy, that a confocal has a pinhole excitation and emission. You know, when you excite and point scan, you are shining throughout the sample. Okay, so the photo bleaching, not, you can answer like high, low, high, middle, low. Okay, photo bleaching for confocal will be
high.
Great. While imaging speed is point by point slow. So what about two photon? Not again. So now excitation light is a IR, near IR light. And so that's a little different. And you're shining near IR throughout too, right? And but two photon excitation happens only at the nonlinear spots. So photo bleaching will be high, moderate, low. High, moderate, low.
I think moderate.
No, you can choose.
I choose moderate.
Oh, very good. Because confocal, it shines throughout. Two photon also shines throughout, but two photon uses near infrared light, so which causes a little less photo bleaching. Okay. But again, it's point by point scanning, so it's slow. But now it comes out light sheet microscope, and you are shining only through this light sheet. You are imaging the whole thing, not PMT pinhole, but the camera you detect. So photo bleaching is you are shining here and you are taking image. Not again, photo bleaching will be?
Low.
Exactly. What about imaging speed?
I get it's fast.
Exactly. Why? Because you are taking the whole thing at simultaneously. Let's say this one point by point, this one, let's say 10 pixel by 10 pixel, this is already 100 times fast, right? But usually you are not only 10 pixel, hundreds of pixels. So even sometimes 1,000 times faster than the other. Okay, so you see the power of light sheet microscopy, and this a nice comparison from Nature Protocol paper. And this one, because of that, you can take cleared brain sample even like 5 millimeter, which composes mouse whole cortex and even hippocampus, or all the things. normal confocal microscope, it'll take a day. Now with this microscope, it takes only an hour. So that's the difference. All right. So for that, this one is reference. So reference means just for reference for your future. I put some consideration of light sheet thickness, which is made out of a Gaussian, and your Z resolution. So from this picture, really important aspect is your light sheet thickness now determines most likely your axial resolution. Because conventional axial resolution is poor, if your light sheet thickness is thinner than axial resolution, because only light signal happens from within the sheet, your axial resolution now depends on the thickness of the sheet. So that's the point, how you can get pretty good, potentially, axial resolution. So those who want to know more deeper, I put this slide. So again, the reference. So all the time, whenever you have a new modality, you want to compare with the other. So there's a nice, you see, Stelzer group made an optical paper to compare with different objective lens and numerical aperture, fluorescence microscope, conventional, confocal microscope. You see two HANU, this is photon energy. So 2 photon fluorescence and your spin, selective plane. So there's all charts. So whenever you are curious about what are their full width halves maximum of lateral resolution, axial resolution, and even the volume of the point spread function. So now we've covered all of the basic concept, and now we get into a little more a practical issue. So far, it sounds like this is great. This solves every problem, but you know, the tissue itself, when it's become more scattering tissue, when you shine it side, tissue element can actually scatter light or absorb light, which makes an artifact. So for example, here, when you shine in one side, you actually get a nice illumination, but as the light propagates, the other side gets actually more dimmer. So an idea is, okay, I shine one time here, take image, and shine the other side, so that like I also want to reveal the right side. And then why don't we just combine them? So that's a nice way to solve. So it's called dual-sided illumination. Okay, so dual-sided illumination make it more even excitation. So that's good. You solve the problem, and the good thing is you just need a half number of rotation, because typically light sheet microscope, while you are shining your sheet, you're actually rotating the sample like CT scan. Okay, but consider aligning both illumination lens to take image and focus. Would it be easy or difficult? Sadaf?
It will be difficult.
Yes. You have to always think a realistic way. Like think you are developing and aligning this microscope. In fact, this alignment is not that trivial. So just don't be fooled by the nice promise this new technique says, all right? And similarly, of this concept, when we take image, a single slice, so this is a zebrafish 2 days old, so it's almost transparent, but still scattering. You see single slice has this, and when you shine illumination on the right, you only shine here, the other side look dark. Hey, that's a problem. So one side illumination, you take one image, and you rotate, you take image, so that it's better to rotate and shine on the other different angles than single slide versus dorsal half. With the multi-view, now you can have much more uniform, nice picture. So that's a trick. Multi-view fusion is necessary. That means you also need to do a lot of image processing as well. So for reference, how to create this, you have this illumination arm one side, but the other side, another illumination, and also detection in both sides. You are taking this simultaneous and multi-view microscope. But this is cool, but you can also think of this, hey, this expensive camera needs to be twice. Objective lens is the most expensive part of microscope. You need to have both. So you gotta be rich to realize this. Okay. And now let's see a little bit how to create the light sheet. So this is a conventional light sheet microscope. For look at that, your 3D samples here. When you create using a light sheet, using a cylindrical lens, what you can see is, look at that nice sheet cut through the sample. And orthogonally, you're taking this image, whole image with your camera. But the issue is that the creation of the light sheet is a little bit like kind of Gaussian profile. It's not always ideal. So another good way is making a line, and you just scan the line ups and down. So this is how you do it, but you need a scanning mirror and so-called f-theta lens, tube lens, to make a low NA objective and line, and you move ups and down. So these two are basically an idea, and the downward one is the second part of setup for light sheet microscope called DSLM. But an advanced one is, now if you scan the light sheet, then you have a control of the shape of the light sheet, because you can scan the sample like this continuously, but you can scan one and then having every other line. So that sounds almost like structured illumination. So yes, that's a structured illumination, which can have an advantage of lateral resolution as well. So this is the concept which also came out Nature Methods of this light sheet. You now not necessarily continuously, but make it every other line, then you can have a structured illumination specimen, which makes you remember structured illumination microscopy. You can increase your contrast and lateral resolution, and this is intensity modulated laser beam. So digital scanned laser light sheet microscopy can be made, and this is very, very cool. All right, so when the light sheet microscopy concept first came out, they're very similar time. It came out 2004 in Science, and similar time 2003, a year before, there was a new technique called the tissue clearing technique called clarity. So people start to think, hey, we cannot take image of big sample because of scattering, but now you can make it transparent. Oh, then you can even make the whole brain with a light sheet. Of course, that's the concept of column from Raju Tomar from Carl Diesroth group from Stanford, who is more famous for invention of optogenetics, but they also do optics. So this is clarity optimized light sheet microscopy. So how can you make it? Hey, let's send the light sheet one side and the same on the other side. And then you put your brain sample and going up, moving up and down to take image. Conceptually, it's a very simple, easy, but in reality, there's the issue of, this is a little bit fundamental issue of light sheet microscope, because you are shining one side, you have detection the other side. So if your detection focus has to be on the light sheet, otherwise you have a misalignment and your image will be blurred. So that is a practical issue, and this is something you can actually use. Each of these you can adaptively calibrate light sheet for, because when you are having an embryogenesis, long-term imaging during the time, you don't want to have a mismatch of focus and light sheet. So that drift is not great. So another one from Janelia Farm made adaptive light sheet microscopy. So each time to finely adjust for the focus. So it allows long-term high resolution. Okay, so far what I discussed is a kind of interesting, But I want to lastly, another new emerging concept, although this one allows a very interesting result, so-called scape microscope, invented by actually my friend Elizabeth Hillman from Columbia University. And this is something interesting. Why? You can actually take a look at a mouse brain and with not the great depth, but a great speed. Why? You look at that. So far we talk about 10 frames per second. But look at that. Hey, this is not F, this is a V. What does it mean? This is volume per second. I mean, that's excellent. Now you can have the whole 3D in 1 second, 10 volumes, which is about 10 hertz. So this, when we talk about confocal two photon, we never talk about volume per second, only frames per second, only one single plane. But now we are talking volume, which means order of magnitude increase in speed. What can you do? Yes, now you can do calcium image, which is a functional neuronal activity. In 3D, you can find 3D branch 1 to 3. All the points you can actually go with a calcium indicator. You can find out simultaneous excitation activity of neurons. So this opens up a new world. Okay. So then how can you build it? So this is reference, but I want to just give you that idea only. Remember when we talk about confocal, how to make it faster? You just scan fast or give a multi-points and scan and multi-point detection. So parallelizing or make a faster scan. In this case, this is kind of a both. Why? You have an objective lens. Now you have a scanning mirror. This is so-called polygonal mirror that while rotating, this can sweep fast. With one objective lens, you are making a light sheet here, like this way, 3D, and you are collecting image of the sheet itself from this point. But like in confocal, you are scanning the light sheet like this. And the problem is in confocal, you can't move your pinhole fast. So what are the trick? You do descan. What it means, when you move this one, you go back to exactly the same mirror. The scanning is descanned and the image remains stationary. So this, what it means is, then you can just put your camera over here, just take picture. And now what's the limiting factor for your scanning speed? Is your camera speed only. And this is a big trick how to make it faster. So optical alignment, where comes that concept? Of course, the concepts are coming from somewhere else, from previously so-called oblique plane microscopy. You don't have to remember, just try to understand. In 3D, there's a 4F system. You have a tissue or sample. You have a light sheet here. Think about that sheet in 3D on the image space. This is a little distorted. Hey, we don't want distorted. How to compensate for that? Oh, wait a second. We can make a symmetric system, so-called a like mirror. So you put this one backside mirroring, then you can create exactly the same one here. Oh my gosh. Okay, that sounds interesting. Now you just take an image with another microscope. Okay, so that's the concept of alignment, and that's how the Cornell group did. So you make this one oblique, so that's a little weird. And you have another 4F system. Now make it a little more vertical. Now, okay, good. You have put 4F system to take image of this one. But while this is scanning, the image space here is stationary because it was descanned by the same rotating mirror system, the same angle. So now you just take pictures of that, and this makes a whole lot of orders of magnitude faster imaging possible. Okay, so then what will be the nice thing and pros and cons? You can see it makes us image faster. but using one objective and unconventional way makes it a little difficult because you are imaging like kind of non-uniform sampling grid, which mathematically you can actually convert. And penetration depth this is essentially like confocal single photon. You cannot go deeper than 2 photons. And you are not utilizing the whole objective. So you are having effective low NA. All right, so for this, I want to give you, this is for your reference. Hey, everything is like data acquisition, but there's a real issue of multi-view fusion, stitching, and even deconvolution. And you will be suffering from spherical chromatic aberration, which needs a compensation. After this, you can get this exquisite analysis and data visualization. So at summary, we discussed about the new concept of light sheet and how to set up in a simple way, and the history, which goes back to about 100 years ago. And this opens up a new applications. But the key is how to produce a nice light sheet and compare this one to the conventional wide field, confocal and 2-photon microscopy, and more recent advances such as SCAPE. So these are reading lists, and I post this year, there's a Nature review methods by Stelcher. So this has a lot more things developed, and the others are like seminal fundamental things. And you can also watch this video. Okay. And lastly, Laxman and I think not, you guys are actually presenting your final team project. It's SPIM. So this one become like open, all the protocols are there. You guys just, it's like a Lego block. You can actually practice. Even my previous students made this. Okay, thank you. And I think